The CAPSIT research infrastructure is a high-containment isolator system that has been installed inside the 3CAPS Biosafety Level 3 facility of the Rager Institute. CAPSIT is a fully automated pathogen-in, human-out, lab-in-a-box system for research on and with live pathogens of higher or unknown biosafety risk. It's a generic system that can perform all essential manipulations, like pipetting, incubation, and sample and data collection. It can be used for research on and with viruses, bacteria, fungi, parasites, or cell biology in general. The main isolator is a stainless steel box of 40 cubic meters that was craned in through the roof of the building. To achieve best leak tightness, it has been constructed out of 2 welded half shells that were merged together inside the 3CAPS facility during installation. In the technical space on top of the isolator, the inlet and outlet HEPA filter systems were installed. The robot system was installed piece by piece in the main isolator chamber. The main robot controls the workflow of the entire system and integrates the different devices into one multifunctional instrument landscape. For capacity and fail-safe reasons, the robot system has been conceived as a Siamese twin, with duplication of each scientific instrument. The robot system is structurally and functionally largely independent of the isolator system. Assay plates and biological material are loaded into the system in closed biocontainers through the antechamber of the main isolator. The same biocontainers are also used to remove waste from the system after surface decontamination in this antechamber. At the human robotics interface, the operator can use the gloves to transfer material from these biocontainers to the robot system. Assay plates of studies that are finished can be removed from the robot system the same way. The extensive level of automation allows to perform research 24 hours a day, 7 days a week. To start a study, the main robot transfers the assay plate to the barcode reader. Once the barcode has been read and recognized by the robot software, the assay plate starts its pre-programmed life cycle in the system. The entire life cycle is documented in great detail. Depending on the content and the time of loading, the plate is transferred to a holding position or directly into one of the 4 incubators. Each incubator has 120 plate positions. The climate in each incubator can be adjusted to match the optimal growth conditions of the pathogen, and studies can even be performed at low oxygen pressure. Before the addition of a reagent and the collection of data, the plates can be transferred to the plate delidder and washer station. After the lid has been removed, the contaminated liquid can be aspirated and, if desired, a washing step can be performed. The event-based scheduling software that drives the robotics continuously optimizes the use of the capacity of each workstation and instrument in low and high demand situations. To start a study with a pathogen of higher biosafety risk, the pathogen is added inside the isolator system by one of the liquid handlers. During the course of a study, samples can be harvested or transferred from parent to child plates according to a predefined time schedule. Before the collection of data, a dye or a reagent can be added, and at the end of each study, a liquid chemical is always added to inactivate the infectious content of the assay plate. Studies can be performed in 96 and 384 well plate format. A room temperature dark store is available to keep assay plates with light-sensitive dyes. Whole well fluorescence, luminescence and absorbance data can be collected in one of the microtiter plate readers. Two confocal LED-based high-content imagers are available to generate vast amounts of microscopic images to document the ongoing biological processes. From these images, data sets can be extracted to study and quantify processes that take place at the cellular and subcellular level. Glove ports allow operators to safely interact with the robot system at critical positions without risking exposure to the pathogen that's handled inside. State-of-the-art isolator technology keeps the system at -200 Pascal and offers maximal protection of the operator and environment. The main isolator is connected to a walk-in L3+ lab by means of the peripheral isolator and its antechamber. Through this antechamber, pathogen samples that are stored in the ultra-low freezers of the walk-in L3+ lab can be safely transferred to the robotics system and vice versa. This small isolator can also be used as an extension of the main isolator or the walk-in lab, or as a standalone unit for the manual handling of samples. Depending on the needs, it can be temporarily equipped with different instruments. All 11 compartments of the isolator system can be individually decontaminated, while other parts of the system remain functional. The decontamination is performed with chlorine dioxide gas, as it can reach any corner of this complex instrument configuration. On the inlet side, conditioned air is filtered twice through two serial plenum H14 HEPA filters before it enters the isolator system. On the outlet side, any aerosols are filtered out by two consecutive H14 HEPA filter cartridges. These are installed in push-push filter units on top of a filter exchange box. This allows to safely replace the filters while the system is fully operational and to decontaminate them in the antechamber of the filter exchange box before removal. Inside the isolator system, the robotics system automatically adds a decontamination liquid to any liquid waste that's generated by the plate washers. This liquid waste is automatically transferred to a stainless steel external waste tank. This tank can be replaced after decontamination of the connection with chlorine dioxide. The content of the tank is thermally decontaminated in one of the 3CAPS facility autoclaves. This makes the robotics system independent of the waste handling and guarantees continuous operation. The same concept is applied to the solid waste that's removed from the system in biocontainers after chlorine dioxide surface decontamination. The software of the Capsid system is designed so that multiple and different studies can be pre-scheduled and performed at the same time. Failure of an instrument merely creates a workflow bottleneck and not a whole system failure. This allows to postpone technical interventions while ongoing studies can still be finished. Medium to high throughput screening and research can be performed in phenotypic assays with live pathogens of higher or unknown biosafety risk, during which, independent of time, multi-parameter data are collected on a large scale.